Welcome back to the session on interactomics. In the last lecture, we discussed about different traditional methods to study protein-protein interactions like yeast to hybrid immunoprecipitation. We moved on to talk in more detail about latest high throughput technologies like protein microarrays, how it can be used. Today, we are going to talk in the same continuation about interactomics. Then I am going to give you an overview of some new tools which can be used like label free biosensors, especially surface plasmon resonance SPR based biosensors, how they can be used to study biomolecular interactions. And then finally, we will briefly talk about systems biology approach, the new emerging field in looking at the high throughput data. Interactomics is a field which is aimed to study the interactions and their consequences between different proteins and other cellular components. The network of all such interactions, which is interactome, aims to provide better understanding of the genome and the proteome functions. So, as shown in the slide, the proteome is very dynamic, and if you look at the connection with other biomolecule is very intricate. For example, it is connected with the genome, metabolome, transcriptome and the various environmental factors. The fluxes of all of them are actually governing the dynamic proteome. And to study proteome, you have to look into the protein interaction networks as well as the protein modifications which are happening like different type of PTMs, for example, phosphorylation, etc. So, all of these are the complex processes involved in governing any physiological systems. So, interactomics aims to identify the function of uncharacterized proteins, trying to define the new roles for the characterized proteins, what are the mechanisms to regulate the protein activity, and what are the possible networks of these protein interactions. So, some of these are the major goals, and to do that people look for different type of interactions which are sometimes the interactions could be very transient just for fraction of second, or could be permanent, which could be very strong interactions. So, interaction could also be divided into the weak interactions or the strong interactions. It could be obligate or it could be even non-obligate. It could be homo-oligomer or multiple units can come together as a hetero-oligomers. So, different ways of interactions may happen, and what could be the physical reasons for such interactions to happen. So, for example, electrostatic interactions, steric, hydrophobic, hydrogen bonds, all of these are actually involved in these kind of interactions, and again studying and knowing that different methods of interactions, what are the physical reasons for them, and then how to use technologies to study interactions becomes very crucial. So, protein interactions, as we have been discussing, that it is quite crucial for any event which is happening in the cell, whether you term as a signal transduction, look at the DNA transcription, translation, even replication process, cell cycle control, various type of metabolic hubs, as well as the morphology and the splicing, the growth of individuals, motility. Many of these things are involved. Many of these biological events actually depends on these biomolecular interaction events, especially protein-protein interactions to happen. So, we have discussed that there are two broad categories to study the protein interactions, and some of the latest technologies which includes protein microarrays and label free biosensors are what we are discussing in more detail to really illustrate all of you that in which way now technologies are really able to help us, the biologist, to find out the information in much more dynamic, in much more high throughput and probably much more precise manner as well. So, today I am going to talk to you about label free biosensors, the label free techniques, how they can be used to study biomolecular interactions. So, here I am providing you an overview of label free techniques, and the image shows at least the 6 of the popular platforms which could be not only label free, but also could be linked to the even microarray based platforms. Among them, the shown is carbon nanotubes as multicolor Raman labels. The B panel shows to you is the planar wave guide arrays. The C panel is about nanowired sensor arrays. D panel about surface plasmon resonance. E panel on the nanohole arrays, and F panel on spectral reflectance imaging biosensors. So, all of these different circles shown to you is essentially based on some physical principles involved in looking at the biomolecular interactions. For example, when an antibody is immobilized on the nanotube, And if a protein binds to it, what is the change in the conductance which can happen, or what is the change in the reflectance in a medium, what is the change happens looking into the interference properties. So, some of these properties have been used to look at the molecular interactions in the label free manner. So, here you are not adding the Psi 3, Psi 5 or fluorescence labels the way we have done in the microarray technology, but rather we are looking at how two biomolecules could interact together, and can we measure that is there any change in the physical properties of them happening, which could be measured using different type of physical based principles and the technologies which are associated with them. One of the platform which is quite popular in the field is surface plasmon resonance or SPR. SPR measures changes in the refractive index of the medium, which is directly in the contact with the gold surface, and as you are measuring the percentage reflectivity change happening, or how much the change in the reflectance is there, based on those measurements you can monitor that whether a binding event is happening or not, which could be measured in the form of sensorgram, which measures changes in the SPR signal versus time. So, for example, as you can see in the plot, on the x axis we have the time scale, on the y axis we have response units, and then we are looking at from the baseline, initially when there is no interaction happening, the stable baseline is there, and as soon as the two molecules start interacting, from those molecules you can see now the association rate can be seen in the green curve. If you are seeing now the binding can be seen. After some time this binding gets stabilized, you can see stochastic steady state signal, which is RU after and before, and then as the binding progresses, you are continuing your experiment. You can if you are floating more of the buffer and an interaction is not very strong, then slowly the molecule will start dissociating out, and that is your off rate. The molecules are getting dissociated, and then finally you can use some mild acids to regenerate the chip, and now that is known as the regeneration process. So, in this manner you can see the sensorgram, which involves the stable baseline, the association or the on rate, the dissociation or the off rate, and then coming back to the regeneration to make the chip, which can be again used back for the these applications. However, the currently available SPR platforms can maximally use maybe 4 channels or the 4 interactions to study simultaneously, whereas think about biological problems when we want to study maybe hundreds if not thousands of these kind of phenomenon to happen simultaneously. So, SPR imaging is a new platform which has come forward, which has ability to combine the power of both SPR and the microarray based platforms, where intention is to look into the high spatial resolution, which could allow for the high throughput analysis of the biomolecular binding. Here you are measuring the simultaneously the entire area of the chip surface, rather than looking at the very specialized localized area what you measure in the SPR. And to do that, the whole chip surface is radiated by the light. Then you are imaging onto the detector arrays, and then you are linking that with a CCD device to capture the whole image. So, this platform has shown a lot of potential to combine with the microarrays. SPR could also be combined with another powerful proteomics platform, which is mass spectrometers. For example, you are looking at how two molecules are interacting, and from your experiment you can see there is a binding event is happening, but you do not know this molecule which is bound what this protein is, right. So, now to identify those proteins, one could elude them out and then analyze them using mass spectrometers. So, now you can say that protein X binding is happening because of the interaction with protein Y, and now you can identify those using mass spectrometers. So, this is a new application which is the SPR MS based application. So, what I want to emphasize you that in which way technologies, integration of technologies, more robust platforms which are coming forward are really helping us to build these kind of newer approaches, which can be so relevant to study this biological phenomenon. So, while we have studied at least in some detail the basics of the label free technologies, we are going to provide you some more detailed understanding of one of the label free biosensor platform, which is surface plasmon resonance. So, now let us have the laboratory demonstration for the SPR.
Hello, I am Dr. Vaishali Kerekate. I am a scientist in Dr. Sanjeev Srivastava's lab at IIT Bombay. Today I will be talking to you and showing you surface plasmon resonance. So now let me show you the sensor chip that we use for immobilizing the ligand. There are many types of sensor chips depending on what kind of molecule you want to immobilize and study. So typically for proteins there is a type of chip. If you want to immobilize a lipid molecule like liposomes, there is a different kind of chip. For today the experiment, the binding experiment we are going to conduct is going to be studying beta 2 microglobulin antibody, which is the ligand, and beta 2 microglobulin protein as the analyte, which will be passed over the ligand. So we are going to immobilize anti beta 2 microglobulin antibody on this sensor chip. This is a CM5 sensor chip, which is a very versatile chip that machine. This is where we conduct our SPR experiments. Let me show you the different parts of the machine. So this is where the sensor chip goes, this compartment. So you control it through the monitor. If you say eject chip, it takes about a minute for it to eject. So you will see it coming out and then you insert your sensor chip. This is the sample rack compartment. This is where your sample rack goes in with all your different tubes. Now if you see on the left side, this is where you have a bottle with your running buffer. So this is the buffer that goes through the machine as well as the one that flows over your sensor chip. This is also typically the buffer that is used for your binding reactions. So you have to make sure that this buffer matches exactly with the buffer that you use for diluting your ligands or diluting your analytes, etc. Then on the right side you can see there are two bottles here. One is just plain water which is used for flushing out, and this is the trash bottle where all the waste comes out. Okay, so this is where this is the compartment where your chip goes in. You can see any kind of chip that you can enter into this compartment. So typically when the machine is on standby, we put in the maintenance chip. So I'm just going to close it and say dock chip for right now. This software that we control the machine through is called the Biocore T200 control software. There is another software here which is called the Biocore T200 evaluation software. So that is where you get the output of your So this is the step of docking the chip. In an SPR experiment has three major steps. The first is the immobilization step. This is where you immobilize your ligand onto the chip surface. The second step is the interaction step, where you are actually measuring the interaction between your ligand and your analyte. And the third step of an SPR experiment is regeneration. This is where after your analyte has bound, you want to make sure to remove every molecule of analyte that is bound to the ligand, so that you make the chip surface available for the next round of experiments. So your ligand stays bound, but the regeneration removes every analyte molecule that is bound in the previous experiment. So now in the first step, we are going to do the immobilization step. We use something called as amine coupling, in which you are using EDC and NHS to activate your chip surface. Your chip surface has a window opens up. Here, each sensor chip has four channels on it. They're called as flow channels. And typically, per experiment, you use two flow channels. One is called a reference flow channel. The other is called a experimental one. So what happens is, in your reference flow channel, you don't put any ligand. It's either a dummy ligand, or you just put buffer through, but it goes through all the steps that are involved in immobilization. The reason being is the reference flow cell allows you to measure any kind of non-specific binding that might be happening. So, and then in your second flow cell is where you include the ligand. So the flow cell 1 goes through every single step that your flow cell 2 will undergo, except that it will not have any ligand on it. Your sample compartment temperature is also 25 degrees. In this machine, you can control the temperature from 4 degrees to 40 degrees. So depending on what conditions are optimal for your binding experiment, you can choose the temperature. Then you click next. So this gives you the rack positions. So basically, it tells you these are the different types of reactants that you will have to put into the machine, and this tells you where to put it in the rack. So if you see the rack positions, this exactly corresponds to the rack here. So for example, this is how you place it. So for example, this position is A1, which is shown here. So this one corresponds exactly to this one. So as you can see, B1 is the description of what you need to put in B1 is over here. So B1 is EDC, NHS, those are the activating reagents. B3 is empty, because these are being very reactive compounds. They will mix inside the machine. The machine will take in the reagents from each of these tubes and then mix it here. So you need to keep an empty tube. Ethanolamine is the reagent that is used to block the sites that have not been occupied by your ligand. And so 1, 2, 3, 4, B1, B2, B3 and B4 are for your flow cell 1, which is a reference flow cell. The same thing gets repeated for your flow cell 2, the EDC, NHS, empty tube, ethanolamine. Here there is one extra tube, which is your ligand, which is the anti-beta-2 microglobulin antibody, that is your ligand. So I am going to place these tubes into each of these corresponding positions. So you have EDC, which goes into B1. You have NHS, which goes into B2. You have MT, which goes into B3. you have ethanolamine which goes into B4. So now we have finished with the B row. Now we start with the C. You get EDC, you have NHS, MT, ethanolamine, and your ligand. Okay, then you close the rack, and this is now ready for inserting into the sample compartment rack. So what you do is then you click on next and place your So now we have placed the rack on the stand, and we are going to insert it into the machine. Once the insertion of the sample rack is over, you click on next. So then it gives you a run protocol, and you have to follow all this. At this point you can check whether you have inserted the correct sensor chip, whether all the samples and reagents are in the rack. So you get this window where you get your run protocol. Here you make sure that at this stage you make sure that the correct sensor chip is docked. Make sure that all the samples and reagents that you want are loaded correctly. Then you place the buffer. Make sure that the correct buffer is in your left hand side tray. Make sure that there is enough buffer for the entire run. Also that the caps are tight and there are no bubbles that will be taken up by the tube. Also, if your waste bottle is full, make sure to empty that. Make sure there is fresh water in this right hand side bottle. So now at the end of the immobilization step of the SPR reaction, we open the Bioco T200 evaluation software, and I've opened the file. The output is a sensorgram. So you click on all sensorgrams. So as you can see, this is, let me show you first the reference cell. So this is the sensorgram from the reference cell. Here, as you can see, this is the step where the EDC NHS is acting, and this is the ethanolamine step where the unbound sites are being blocked. If you go to our experimental flow cell where we have added the ligand, as you can see here, this is the EDC plus NHS. Then the ligand is being bound to your surface, and then as you put the ethanolamine, your unbound sites are being blocked. So you can see the difference between your experimental cell and your reference cell where there was no ligand being bound. So that concludes the immobilization step. Your ligand is now bound to your sensor chip surface. Now we will go to the next step, which is the binding reaction. In this step we will be passing the analyte over the ligand, over the chip surface which has the ligand on it. Now we will start with the binding experiment. Once again we work with the control software. We are going to open a new wizard template. Bind want a yes no answer. Is your analyte binding to your ligand or not? So we are giving very simple instructions here. We give a very standard flow rate. We don't need to do any extra steps. We just want a yes or no answer to whether binding is happening or not. So we give a contact time of 180 seconds. That is enough for your ligand to bind to your analyte. Your flow rate is 10 microliters per minute. Then your regeneration solution, in this case it is a low pH solution, glycine HCl, so the contact time of your regeneration solution on the chip will be 30 seconds, flow rate of 10 microliters per minute, and a 5-second stabilization period. Then you click on next. So your beta 2 microglobulin protein will be in 2 different concentrations. So you can check it under low concentration and under high concentrations, and each of these will be done in duplicates. So the low one and low 2, high 1 and high 2. And then since we have already primed during the immobilization step, we will not be doing that. The temperature settings are the same. Then you go to next. So again you get the rack positions here, and it tells you in which position to put each of your reaction mixture. So the first one is your glycine HCl, which is a regeneration solution, goes into A1. So I am going to place the glycine in the A1 position. Then in your B1 is the beta make sure all your checks are in place. Your samples and reagents are loaded correctly. Make sure your buffer is placed correctly in the left hand side tray. It gives you an estimated run time. So this entire binding reaction will take about 26 minutes. And then you hit on start, which will start the binding reaction. Once again, once your binding reaction is over, you open the Biocore evaluation software, and you can open your sensogram through this software. So let's open the file, click on all sensograms, and here you can see the lower 2 lines are the low concentration of your analyte, and these 2 are the high concentration of your analyte. So as you can see, this initial part is where the buffer is being flown over your sensor chip. As you start, as the analyte starts flowing over your chip, you see an increased binding. At some point, you stop the analyte and you start the buffer, and it starts going down, and then you have your regeneration solution, which gets it back to baseline, and then again your buffer is flowing through. So as you can see, the low concentrations are giving a low binding, whereas the high concentrations are giving a high binding, and the duplicates are showing repeatability. So basically the binding experiment has worked. Let me also show you a kinetics experiment, which we have not performed, but I will show you a typical sensogram that you get from a kinetics experiment. So what we saw just now was very basic yes no kind of experiment where you're asking the question, is my analyte binding to the ligand or not? In a kinetics experiment you get much more information, so it is a it's an experiment where you can find out the association constants, the dissociation constants, the affinity constants. Basically you have a concentration range of analytes that you will pass over your ligands, and the you can calculate the various constants based on the sensogram curves that you get. So let me just show you. So click on all sensograms. Surface bound, because we are measuring kinetics on the surface of the chip. Okay, so here what we have done is we have used a range of analytes. We have used 0, 2 nanomolar, 4 nanomolar, 8 nanomolar, 16 nanomolar and 32 nanomolar of analytes that are passing over our sensor surface. So if you see these curves, this is the 0 concentration, 2 nanomolar, 4 nanomolar, 8 nanomolar which was carried out in duplicates, 16 nanomolar and 32 nanomolar. So as you can see, here you have the buffer. At this position, at this point, at 0, you have started your passing your analyte over the ligand. So you can see an increase in the association of your analyte with the ligand. At this point, you stopped passing ligand and only buffer is passing. So you can measure the dissociation of your analyte from the ligand. Okay, and then you have regeneration. So if I click next, let me just go back for a minute. So here again you are using 2 minus 1. So in SPR it's very important to do something called as double referencing, where you use reference subtraction as well as blank subtraction. Blank subtraction means you always have a 0 concentration which is subtracted from each of your measurements. And reference subtraction means you are subtracting the reference flow cell from your experimental flow cell. Both these ensure that the signal that you are measuring in your experiment is very specific to your ligand analyte interaction and not due to some kind of non-specific binding that is being which is brought about by any kind of molecule in your buffer. Okay, and then this is the output. So if you see these tabs here, there are 4 tabs: quality control, report, residuals and parameters. So in your quality control you see that all of these you see all the green tick marks, which means that all the QC checks have passed. So kinetic constants are within your instrument specs, the kinetic constants are uniquely determined and there is no significant bulk contribution. If you go to the report, you can see that it has calculated your association constant, your dissociation constant and your affinity constant. Your chi-square value should be less than 1, which it is showing. Your U value should be less than 20, which again it is showing. So these are pretty good results here. Your residuals should be within these green lines here. That means you can accept this data. If it lies beyond these 2 red lines, that means you cannot accept this data and you have to go back to optimizing your experiment, seeing what went wrong. But here there seems to be a very good fit. This is what a sensogram from a kinetics experiment looks like.
All of these omics technology, which is kind of high throughput data generation technology, which generates big data in a very short time and very short span of duration, aims towards looking at the entire systems. But that is somehow not possible if you are looking at only one part of the picture, or if you are looking at only the genes, or looking at only the RNA or the proteins. You may not be able to get the entire picture of what is happening in the physiological system. So, system biology is a field which investigates the behavior and relationship of all the elements of a particular biological system, and looks for integration of the data, which could be then eventually computationally modeled, and then finally you can display those information to enhance understanding of a complex physiological systems. So, system biology aims to provide the systems level understanding of biological networks. The biological information which you obtain could be represented in the form of the networks, which are for the interacting elements, interacting proteins and other biomolecules, and how they are dynamic in response to various perturbations which are happening. So, these networks could provide the insights which cannot be analyzed from the individual isolated components of the system, and therefore, you have to pay attention to various components which are equally important for the system biology field, looking at the networks, looking at the models, doing the computational analysis and the dynamic property analysis. Broadly, there are two major approaches which people have used to look at the systems based data, model based or data based analysis. In the model based, the prior information is implemented in the model itself, whereas in the data based approach of system biology, the objective is to find out what is the new phenomenon there. In model based system biology relies on computational modeling, for example, different simulation tools, whereas data based relies on the omics data sets. The model based field is difficult to build detailed kinetic models, whereas data based you are having various information from the experimental data. So, you can look into the complex relationship among genomic, transcriptomic, various type of other proteomic or metabolic networks, various pathways and networks which could be derived from the same. A systems biology triangle involves majorly three aspects. One is the experimental part, where data is being generated. Second is computational modeling, where you are proposing some models. Third is the integration with the technologies. So, all these three are the important components of systems biology, which is known as systems biology triangle. The synergistic application of experiment, theory, technology and modeling to enhance our understanding of biological processes as the whole system, rather than looking them at the isolated level. So, what is important here, that we want to understand the whole system, and if you are looking at systems, probably the system properties are, it is not just addition of looking at the DNA plus RNA plus protein and going to give you 1 plus 1 plus 1 3, but rather these individual component information, when you analyze in the complex network form, you will find that probably a new system properties are emerging, which is giving you better clues and better understanding of the system itself. So, that is what is the power of this new field of systems network and system biology. In conclusion, we have discussed today the dynamic protein molecules which interact with variety of other biomolecules for in the field of interactomics. These interactions could be studied using the label based platform, which we talked the last class, especially the protein microarrays, or it can also be studied in the label free manner, which is looking at the surface plasmon resonance or many other physical principles which could measure the protein interactions in the label free format. The systems level understanding is really crucial to provide the understanding of the complex physiological systems,